Okay, so essentially, when I conducted the study, it was really just to do a chemical analysis, a biochemical analysis of sargassum. We found over 200 compounds, and of the 200 compounds, there were quite a few that stood out in terms of having pretty high retention rates, pretty high error percentage, and pretty high quality scores. And so we took the top 10 of those and we looked at potential areas of application for each. And when we saw that two of the compounds in particular were favorable in PCMs, and we did a further analysis to look at the if research has been done to see if sargassum could be used in PCMs, and we realized that it's a very new area. As mentioned in the presentation, there are only two scholars who have actually looked at it, and it's really last year that they did that research, and the research has been very promising, with the only gap really to look at how to optimize the conversion of sargassum to a suitable form of PCM for its further industrial application.
Right. Okay. All right. Great. Thanks. Some of the things that you said there, wasn't really in the presentation. So I'm really glad you clarified those things. Also, let me just see here. Could you clarify for me how much product did you say get out of maybe 100 gram of sargassum versus, well, how much of the PCM did you get out of 100 gram of sargassum, or maybe 1 kilogram, etc.?
So it will take a little bit of time for me to bring up the numbers, because that's in my thesis paper, but I
I think
do
it
have
was
the conversion ratio. I do have the conversion ratio, which
Lenika, were you
oh,
talking grams per liter of sargassum? Were you doing it that way? Is that easier for you to respond?
no, I don't have the numbers. I have the conversion ratio. So
That's
it's
fine. That's quite okay. I'm glad you addressed it in your thesis, right? So we don't need to to stick too much on
okay.
that
You want
question, right?
figures? Okay, no
Okay, great.
problem. But it was a very low ratio.
Okay, all right. But so in your thesis, have you contextualized that those values with the two previous studies?
Yes, I would have.
Yeah. Okay, all right, great. So you did mainly chemical tests. Were you able to relate the chemical tests to like physical properties of the PCM product?
No, I wasn't able to take it that far because there are other aspects of the study. This is a microfragment of it. We
That's
looked at
fine.
quite a lot of applications. So even bioethanol, biomethane, biodiesel, we looked at. Yeah.
Yes. Okay. So I would like to say, 10 minutes to present your work is really, you have to present only a very focused thing, right? So I appreciate that, right? I appreciate your work is
Thank
going to be
you.
a lot wider than what was presented here. Okay. Right. So by and large, I did enjoy your presentation. It was very precise. There's just one thing here that and I think this goes across for all of the presenters, in that there were times where I didn't see citations to previous studies. Although you all did speak about it, it was not, those citations were not on the slides, right? And just, generally across for all presenters as well, in terms of methods, sometimes you need to sort of like clarify if you are using standard methods as well. Again, so this is where citations to previous studies will help with clarifying that. Right?
Okay, but it was on my slide for the methods as well, and the literature review slide, the
No,
citations.
yours had it, right? But I was just talking across for all presenters, right? But yes, I totally enjoyed this presentation. And again, congratulations on the really good work that you are doing.
Thank you so much.
Dr. Felix? Any particular questions for this presenter?
Actually, you asked the question that I have written down for this presenter. I was really concerned about from your experiment, the the you showed the test tube with that. We were not trying to figure out the units, milligram per liter. So I was wondering now, what is that, the practicality, if that were to move from the lab into a read, if it's practical. So I was concerned about the practicality in terms of real life um,
Oh,
use.
yes. If I may add, so looking at even though the conversion efficiency was actually pretty low, that's usually what happens within the industry when they're using bio-based materials, and so it has been found to still be acceptable, and it came out to be one of the high-ranking alternatives that is found to be suitable. So it's still within that acceptable ratio.
Good. Thanks. That was my main question. Thank you.
Okay, thank you, Lenica. I have some questions for Kian. Kian, I know you had a bit of a technical difficulty, but I must say you held your own in moving through the presentation. You also had some excellent, I must say, very specific objectives and research questions, right? So that really helped to understand some of your work as well. I just have some broad-based questions here, and one is in terms of the production of bioethanol from sargassum, are there external factors that could affect the bioethanol quantities?
Yes, there are. Some of the factors that can affect it is the time of year that you collect the sargassum. As we saw that the carbohydrate content would vary during the year. So it is better to collect the sargassum during the summertime when the carbohydrate content is greater and you stand a better chance of getting a higher carbohydrate content.
Right, so that carbohydrate content that's greater during the summertime, is that related to temperature? Is that related to I don't know, sea surface temperatures or something like that? That's what I'm trying to see what's the link there between chemical content and external conditions.
Okay, so yes, it does. Temperature does affect it. We know that sargassum is a photosynthetic plant, so it carries out photosynthesis. So when the temperatures are high, photosynthesis takes place more greater, and then it has a ability to store more of the carbohydrates as compared to lower temperatures. Also, the age of the sargassum also affects it. So the age of the seaweed, the time you collect it, can affect the carbohydrate content.
Okay, all right, great. I think you said in your results that the average ethanol production was 104 grams per liter.
Yes.
How does it compare with previous studies?
So
Either with sargassum or with other brown type seaweeds.
Okay, so when compared to other sargassum, which is the same sargassum specific, which was carried out in, there was one in Indonesia as well as one in the Philippines, I saw a rate of 19.9 grams per liter and one about 2.79 grams per liter. When compared to other brown species, we saw that there was a rate of over 600 grams per liter seen. And also it is important to note that yes, I may have gotten 104 grams per liter of ethanol, but there was still a lot of the reducing sugar which had not been converted to bioethanol present in the medium. So we know that there is a potential for a greater amount of ethanol to be produced as well, which can compare to the 660 grams per liter.
Right, so to do those comparisons, did you perform the same experimental methods as those studies?
No, so the previous study used only cellulase, whereas I used a variation of a number of enzymes. So that could have contributed to a greater breakdown of the seaweed. Okay. Whereas the one just the 660, this only used acid hydrolysis. There was no use of enzymes.
Okay, all right, great. I saw that in your methods that you did fermentation for about 72 hours. Is there a reason why 72 and not maybe, I don't know, 48 or longer?
Okay, so most of the literature I found, they used 48 or 72. So I decided maybe it's better if I just use the 72 hours to maximize the breakdown of the carbohydrates, the polymers in the seaweed, so I could maximize the amount of the reducing sugar that I obtain.
Right. So have previous studies looked at the effect of the fermentation time, in particular for sargassum?
Not to my knowledge. I know that they have varied the type of yeast used, but not the period of fermentation time.
Okay, all right, thank you. That's my questions for you. Again, excellent presentation.
Okay, thank you very much.
Okay, right. So Kiana, very good presentation, and also I know of your technical difficulties, but you really stood strong through it. So good going there.
Thank you. Thank you.
So yeah, I did enjoy your presentation, and what stood out for me in terms of me asking you this question is, have you considered is there any environmental impact in if you have large scale harvesting of sargassum?
Well, I
For
haven't
your
considered the impacts of large scale collection of sargassum. I know that it does affect, being on the beaches does affect the region, the environment. There are environmental factors of it being there. There are health issues. It affects, but I have never studied the factors in mass collection.
Oh, okay. So that is in the process of still trying to come to do your experiment and get your results and quality, because your project is actually quite interesting. Environmental impact is something as well that should be at least considered somewhere in your
Okay, thank you.
write-up. But generally, it was really a very good
Okay, thank you very much. Thank you very much.
Okay, thank you, Kian. And my next question is for Shamika. Shamika, first I must say, excellent description of your methods. One of the things I appreciated there was that you did 10 replicates for your experiments, and having a larger number of replicates does give more rigor to your final results, right? So I really did like that. There were one or two things I wasn't too clear. So your biomethane aspect is separate from the distillery aspect, right?
No, everything is together. So I co-digested the sargassum seaweed and the rum distillery wastewater, and then I produced the biogas in general, and then I scrubbed it to remove the trace gases so that I could have biomethane separately.
Right. Okay. All right. Sure. One of the things that I also liked was how you linked your results to your recommendation as well, in that the next step, one of the next steps could be microbial testing. Have you looked at the literature on that by chance?
I've looked at it not in much detail. I'm going into that for my PhD aspects of the study.
Oh, excellent. That's nice. Okay, great. Could you differentiate for me how is mono and co-digestion different?
Okay, so with monodigestion, I'll use the example of my experiments. I would just place, for example, sargassum seaweed in an anaerobic digester, and I would produce the biogas from that, adding an inoculum or a bacteria source. But then if I'm doing co-digestion, then I would put two different substrates or types of organic material into the digester. So that would have been, in the case of my experiments, the rum distillate